It is just a huge honor for me today to be podcast interviewing Dr. Herb Moskowitz, is the chairman and CEO and co-founder of ioTech International. He is a co-author of ioTech's patent and global patent applications. He previously served as chairman and CEO of Advanced Tissue Sciences and also Life Medical Sciences, two publicly traded medical technology companies. He has served as a board member of several other public and private medical technology companies. He has previously practiced clinical dentistry for more than 25 years, having graduated from the University of Tennessee Dental School. How'd you get all the way from Tennessee to Boca Raton, Florida?
I just went downhill.
You just went downhill?
We just followed the curve.
So is University of Tennessee, that's in Nashville, right?
No, it's actually in Memphis.
Oh, Memphis. My gosh. What am I thinking? University, I've been there. Unbelievable. So, man, Memphis, I bet that was a fun place to go to dental school at Beale Street.
Beale Street, Elvis, the works.
Yeah.
Yep.
So let me, I want to read your website because I called you, you didn't call me. This isn't an advertisement, but I want to read your company background. ioTech International was founded 5 years ago by a dentist and two chemists who converted a Florida garage into a laboratory. The founders knew of the incredible power of molecular iodine to rapidly destroy a broad spectrum of pathogenic microbes. They also knew that, as deadly as iodine is to germs, it is amongst the safest of all substances to humans. So safe, in fact, that it is actually an essential nutrient necessary for our health. For decades, scientists have been confronted with a paradox that for 50 years has been unsolvable. Water-based iodine antiseptics
I have to move that.
Are used globally for their superior germ-killing properties. However, the only biocidal germ-killing form of iodine is molecular iodine. For every atom of molecular iodine in these antiseptics, there are over 10,000 atoms of other types of iodines, which have no capacity to kill microbes, but contribute greatly to staining and toxicity. The paradox, simply stated, is that which is present in least supply contributes to greatest efficiency. Our intrepid band of founders toiled mightily for 3 years to develop stable molecular iodine formulations that boost the active molecular iodine concentrations by more than 100-fold, while reducing the non-active but toxic forms of iodine to a mere fraction of their former presence. Having successfully developed the underlying core technology with a U.S. patent granted and with patent applications pending globally, the company moved into larger quarters suitable to support its multiple international initiatives. Dental products based on this innovative technology have been tested and have been received best-in-class ratings from a well-known and highly respected independent dental research organization. ioTech's technology is now being introduced into agriculture, medical, and dental sectors. Within the dental sector, products are being developed for use in periodontal treatment, caries control, wound healing, infection control, topical antiviral, antifungal, antibacterial. The reason I want to get you on the show is that, when you and I were little and just got out of school, periodontal surgery was all periodontal surgery, and then the pendulum switched to extracting those periodontal teeth and placing implants. And now, 30 years out of school, they're looking at all those implants and saying, my gosh, after 10 years, probably almost half of them have peri-implantitis. Is this something you think will be used for the treatment of peri-implantitis?
It's actually being used right now for that purpose. And this is a very, very important application. So, in fact, it's so important to us that we felt that this was the first product that we wanted to develop from this ubiquitous technology, which can literally derive hundreds of different products from the technology. So this is in all different fields, but particularly in dentistry, for professional dental offices, and particularly for periodontal applications. We see an immediate and an important application. So yes, and in fact, it actually runs the spectrum from helping to prevent to helping to treat. And so, and this is these are for people who are at the very incipient stages for gingivitis, all the way up through a severe periodontitis. So yes, absolutely. And when we're done with periodontitis, and as you said, now we're into peri-implantitis. So it's a very important application.
Well, you're going to have to walk us through the journey. How do you go from University of Tennessee in Memphis for practice clinical dentistry 25 years to actually discovering this new technology? How does that work?
Yeah. well, I actually practiced in upstate New York, in the Albany, New York area for many years. And while I was practicing, I was introduced to a young technology in the medical space. There were a couple of medical researchers who also were developing some technology in their garage, actually. And when I looked at this technology, I thought it was so profound and so compelling and they needed so much help that I pitched in initially on a passive basis with financial support, and then actively, because they needed that much help, and it really was at a point that if I wasn't there to help them with that, then the technology never would have seen the light of day. So it was at that point that I actually left dentistry to help to develop this technology. That technology was the beginning of the stem cell revolution, and that technology wound up to becoming dermal grafts, where living human dermis, sheets of living human dermis, were grown up in the laboratory, and they were used initially for a replacement for animal trials or animal testing. So cosmetic manufacturers, pharmaceutical manufacturers who needed to do animal testing and then human testing before their products could be approved for use. If we could obviate the need for animal testing by providing living human tissue that they could test safely in the laboratory on, then they wouldn't need to test on animals, because ultimately, even if it works on animals, they still have to try it on humans. So this was a safe and effective way of testing on living human tissue without endangering a human being and without endangering animals. So that was a series of products that we brought to market. And then also, we also grew up sheets of dermis and had them cryopreserved in burn centers around the country, so that when someone came in with third-degree burns, a surgeon could just lift the sheet of dermis out of the liquid nitrogen and apply it to the patient. And the beauty of human dermis is that it does not have antigenic factors, so that there is no rejection of human dermis. The rejection factors lie in the epidermis. So once this sheet of dermis was applied to these wounds, then the patient would not need a second wound to harvest tissue for a graft, because they were using this donated dermis which had been cryopreserved, and then it was just seeded with the patient's own epidermal cells, and it actually grew back beautiful skin. Beautiful skin. So that's being used now in burn centers around the country. And then similarly, for non-healing wounds, venous stasis ulcers, decubitus ulcers, these wounds, there's been applications that have been found for the same tissue to be used to help to heal those wounds. So that was my initiation to what brought me out of dentistry, because the technology was so fascinating. And once I had done that, then I was looking at other medical applications to develop, and we found the case of MRIs. We developed a site-specific MRI scanner, which is as small as a dental stool. And that site-specific scanner allows for very, very inexpensive MRIs, where a patient can have an MRI. The MRI can be in the corner of a doctor's office, an orthopedist's office. So it could, if it was for a limb, if it was for a knee, if it was for a wrist, then those applications can be readily done right there in the doctor's office, and done very, very inexpensively, that MRI scanner we brought to market. Then, in addition to that, we also developed a treatment for surgical adhesions, or actually for the prevention of surgical adhesions, and particularly for cardiac adhesions, because there's always been a problem when someone goes in for cardiac surgery that cardiac surgeon will tell you, if you want to see which was an operation as compared to a reoperation, just look on the ceiling of the surgical suite. And if it's covered with blood, then you know that it was a reoperation. Because in order to be able to get to the heart, then they have to tease away all the surgical adhesions that had formed from that first operation. So within those surgical adhesions, blood vessels, arteries and arterioles also form. And so it means adding additional risk for the patients of several hours more on the table, and also the additional suite time and surgeon's time. So in its stead, we had developed a biopolymer, which can be, it looks much like saran wrap, but it's bioresorbable and can be tacked to the inside of the chest wall. And it helps to prevent surgical adhesions from forming between the heart, the pericardium of the heart, and the chest wall. So these adhesions form within the first 2 weeks following surgery. So if the biopolymer resorbs after a few weeks, so in that interim period, the adhesions are prevented from forming between the chest wall and the heart. And so that when a patient goes in for a reoperation, then the surgeons don't have to deal with that additional operative time and additional risk to the patient. So that product was approved in Europe, and it was also approved in the United States. So that's an additional product. Also developed a product for scar remediation based on silicone padding and silicone gel, and those were brought to market. And we also developed a device called the Sure Closure device, which is a one-time use disposable device, which helps to convert tissue deficit wounds that are non-suturable. They're so large because of the deficit of tissue in there. Perhaps it's because of a traumatic wound, or because it's cancer surgery where a good deal of tissue is being removed, where the borders of the wound are so far apart that it cannot be sutured. So it requires a fill-in with granulation tissue, or it requires a graft. In its stead, these massive wounds were able to be closed with this device, which actually, using a tension over a period of about 20 minutes or so against the borders of the wound, allowed these collagen fibers, which are randomly aligned in the skin. And this is what's known as the viscoelastic properties of skin. So these randomly aligned collagen fibers with tension, controlled tension, line up and slide past each other, so you get an enormous amount of stretch, and so that these wounds can be sutured. So what we used to require a 2-week stay in the hospital with grafting and the creation of a secondary wound for the harvest site of the graft no longer is necessary in many cases with the use of this device, because that same wound can now be sutured in 20 minutes or so. Amazing device. In fact, we first saw this device. It was being used in Israel because of traumatic wounds. And I saw one soldier where shrapnel had blown through his leg, and the wound was as large as a grapefruit. And that wound was closed with this particular prototype of this particular device. So that was a product that we got onto the market, FDA approved, and that's out there helping people. So along this process, we actually evolved to what I think is probably the most relevant and most most ubiquitous opportunity, which is this iodine chemistry. And this iodine chemistry is something that I had come across with in a small company in Massachusetts that I had helped to fund while it was still in its startup stage. And that company had developed a high potency iodine technology. And that iodine technology was used to create bovine teat disinfectants, a new class of teat disinfectants that were much more effective to help to prevent bacteria from getting into the human food chain when cows are milked, so that the cow udders are disinfected first with this particular iodine treatment. And that was marketed to actually in about 40 countries. So the USDA requires that cow udders be disinfected before they're milked to go into for human consumption. So that company made a breakthrough in that it developed high levels of iodine, but they were not able to keep that technology stable, so that after a few hours, the iodine would dissipate and the effectiveness would be gone. So it was at that point, because I had been a member of the board of that particular company, I had intimate knowledge of the effectiveness of high levels of iodine, and I subsequently I was talking to the chairman of the Department of Otolaryngology at a major teaching hospital, and he was telling me that they had done some studies where they developed an antiviral as a throat spray, and that antiviral, they used it for viral pharyngitis, for sore throats, virally induced sore throats, and they felt that the results were so good that they felt that it was curative. So this was it was an initial pilot study, but it got me to thinking, because I already knew about the important uses of iodine in dentistry. And I thought, oh my gosh, if they can treat topically the viruses that cause sore throats, and it does not have to be treated systemically, and the way it was explained to me by this ENT surgeon was that the viruses burrow into the mucosa of the throat, but they're still accessible topically. So with a twice-a-day throat spray, they were actually getting effective results. And they were doing that with a plant-based extract, which was similar to like eucalyptus and other aromatic oils which are extracted from plants. And at the time he was telling me about this, I already knew about the tremendous efficacy of iodine, and that how it is so much more effective as an antiviral. In fact, it's unbeatable. So we went back to the drawing boards. And even though this first iodine company that I told you about, they had 3 PhDs working for 4 years trying to get their iodine to be stable so that it could be used in commercial applications other than as a bovine teat disinfectant. But they were unsuccessful at it. So we thought, you know what? This is a long shot. It probably will not work. We probably will not be able to develop it. But we went back to the drawing boards, and we actually spent 3 years trying to get a high potency molecular iodine formulation which would be shelf stable, that could be used for hundreds of different germicidal activities, for agriculture, for healthcare, for industry, for consumer products across the board. And lo and behold, we were able to do it. And we had on our team, we have some really expert iodine scientists. And they were able to come up with a formulation, and a stable formulation that we've shelf tested for 2 years or more. So we have products that we can use in these applications. And the beautiful thing about iodine is that it's an essential nutrient. it is so safe that our bodies require iodine in order to prevent iodine deficiency diseases. And in fact, so that's kind of what led me into our forming of ioTech. And we have a patent on that technology in the U.S., and there are patents pending worldwide for that same technology. And there's the huge product opportunities in a number of different areas. So that's kind of how we went from practicing dentistry to developing iodine formulations.
Wow, the halogens on the periodic table, they're really common in dentistry. I mean, you got on column 17, fluorine, chlorine, bromine, iodine, acetine. How do you pronounce that? Acetine?
Acetine?
Acetine, the column on the periodic table. Underneath, going down, fluorine, chlorine, bromine, iodine, acetine. That is amazing. It's an amazing atom. It's an amazing column on the periodic table, the halogens, aren't they?
Oh yeah, and that's why they have widespread use for these germicidal applications.
So,
yeah, go ahead.
So on all these previous technologies you've invented, where are they going into dentistry now? And I see you have B2B, and you also are going after the B2C. You kind of remind me of Apple, Steve Jobs starting this thing in a garage. And my gosh, and then not only that, but you remind me of Bob Ibsen at Denmat, who started what was his toothpaste called? The Rembrandt. And the mouthwash,
yes.
Launching a B2C product, that's a totally different world than B2B, business to business, dentist to dentist. But going into the B2C world with your iorinse mouthwash, this is kind of are you the reincarnation of Bob Ibsen? Is this mouthwash going to take off?
I don't think so, because it's not our intention for us to go B2C with the iorinse mouthwash. What we're developing now is a group of products for professional dental use only, for use in dental practices and on the recommendation of dentists. These products are not sold in stores, and we don't intend for them to be sold in stores. Now, having said that, there's separately there will be a line of products which will not be the same as the ones we're marketing to the dental community, but they will benefit from the same core technology. And those applications we intend to out-license to major players in the consumer products world, in that particular case, and other major innovators that already have established infrastructure in their various respective markets. But we ourselves do not intend to develop and market to the consumer space.
My gosh, if you were on Shark Tank, the bald, beautiful Mr. Wonderful would love you. That's what he's always telling people on Shark Tank. There's already major players in this space. Let's license the technology to them. Let's not reinvent the wheel. So I think Mr. Wonderful would be in love with you.
Yeah.
Have you seen that show?
I've seen it from time to time. Not enough to know who Mr. Wonderful is.
He's the bald one from Canada.
Okay, got him.
You'll notice in any group, the bald guy is always the smartest one.
Right. Right. Well, there you go. I'm looking at one right now.
So tell us more of your dental journey. So how is this going to work? The dentists listening to you right now are commuting to work who have, gosh, if you're a periodontist, you probably have hundreds of patients with peri-implantitis. What would you be telling them to do right now?
Okay, so in order to get a good grounding in this thing, is to understand really how do we differentiate the products that are out there now compared to the innovations that we're bringing to market. So then I think it would be useful if we can just take a look at what sets this technology apart and why is it so superior to existing technology.
So you're going to do a slideshow right now?
I'm not going to do a slideshow, but there are 2 or 3 slides which can
Okay, so if
which
you're
can help to point that out.
So if you're listening to this on iTunes, switch over to YouTube. It's youtube.com/Dental Town Magazine, where we have all 1,200 podcasts. Thanks to the 9,500 subscribers on our YouTube channel. We appreciate that immensely. But yeah, so switch over to YouTube right now because Doc's going to show us some slides.
Okay. All right. So probably to get the best benefit of this, I think it's important to know that iodine has been around forever. In fact, the first recorded use of iodine has been 5,600 years ago by early Chinese physicians. They used iodine, or they used products which contained iodine to treat goiters and large thyroid glands, and they did it successfully. And they did that by grinding up seaweed and sea urchins and sea sponges and making a paste of it and wrapping it around patients' necks. So this was all done before iodine was even discovered as an element. They didn't know, the early Chinese physicians, that they were using iodine. They knew it worked, so empirically it worked. It wasn't until the 1800s that iodine was discovered as an element, and the first types of iodine which we used therapeutically after its discovery were alcoholic solutions of iodine, tinctures of iodine more particularly, because iodine dissolves very, very readily in alcohol. It is only sparsely soluble in water. So the first solutions of iodine were tinctures, alcoholic solutions, and they were very effective, and they are very effective. But they didn't get as much traction as they could because of the alcohol. Then anytime it was put on an open wound, then the alcohol burned and stung. So it had limited utility, even though it was recognized as a very effective antiseptic. It wasn't until the mid-1950s that iodophores, particularly povidone iodine, which is a water-based solution of iodine, was introduced. And these water-based solutions, even though the iodine molecules were not soluble in water or barely soluble in water, they introduced polyvinyl pyrrolidone, a large organic molecule which is extremely water soluble, and it physically bonded. That molecule physically bonded to the iodine molecules and dragged the iodine molecules, kicking and screaming, into solution. So they were able to develop. In fact, this was done at NASA. They developed water-based solutions, and this has been in use for ever since. So water-based solutions of iodine, povidone iodine, betadine is a common name, have been in use continuously since the 50s. And they're used around the world. They're used in hospitals around the world as surgical scrubs. They're used as preoperative patient skin preps. But there has been no major innovation in iodine since the introduction of iodophores in the 50s, until the advent of our technology. So this is really the most important advance in iodine chemistry in the last 60 years or so. So if you compare povidone iodine to our technology, you'll see that first, povidone iodine is, we call it iodine, but it's actually a gamish. It's a mixture of several different species of iodine. And of all those different species of iodine, only one species is bioactive. And that biocidal species is molecular iodine. And molecular iodine is only present in povidone iodine at 1 or 2 parts per million, a maximum of 3 parts per million. The rest of the iodine in povidone iodine, which is hypoiodous acid, triiodide, iodate, iodide, Those iodine species constitute the vast bulk of the iodine present in povidone iodine. But those species of iodine are not bioactive. They don't kill germs, yet they contribute to toxicity and staining, right? So in povidone iodine, you wind up with about 31,600 parts per million of all these different species of iodine, and yet only the 2 or 3 parts per million of molecular iodine do all the heavy lifting. They're the ones that do the killing of germs, not the other species. So for our purposes, the rest of the iodine is all garbage iodine. It just has negative consequences and does not contribute to biocidal efficacy. We have been able, through our technology, to develop formulations which are 1, 2, and even 300 times as much molecular iodine, yet reducing the other species of iodine to just negligible quantities, so that the total amount of iodine in our formulations are just perhaps 8, 9, or 900 or 1,000 parts per million total iodine, compared to the 31,600 parts per million in povidone iodine, and yet it's hundreds of times more effective. Okay, so if you look at this diagrammatically, for those of you who are able to pull up your screen, you'll see that the diagram on the left is schematic, represents 10% povidone iodine, which is what's used in dental offices and hospitals, and most of the molecules are not molecular iodine. There's only 1 out of 10,000 atoms of iodine are the molecular iodine atoms, whereas our iodine in the diagram on the right is predominantly molecular iodine and very few of these other species. And this next slide shows a graph which shows the concentration of molecular iodine in povidone iodine, and 10% povidone iodine, which is currently used in dental offices, contains at most 2 or 3 parts per million, as you see on the screen. When you compare the efficacy of povidone iodine to our molecular iodine formulations, there was one study that was done which utilized poliovirus, which is a non-envelope virus which is extremely difficult to kill. And 10% povidone iodine solution took 15 minutes to achieve a 0.6 log kill. 15 minutes for a 0.6 log kill. Whereas our solution was able to completely inactivate poliovirus with a 4.5 log kill in 90 seconds. So there's a big differential in the actual results compared to povidone iodine and our molecular iodine formulations. So compared to both chlorhexidine and povidone iodine, the ioTech formulations are stronger, they're faster acting, they're less toxic, they don't irritate, they're non-staining, and they have a much broader spectrum of activity. And in fact, when we talk about activity, we had compared against a very resilient fungus, Aspergillus braziliensis. In fact, that's used by microbiology laboratories as kind of the gold standard. If you can kill Aspergillus braziliensis, you can kill any fungus. And we tested it not against chlorhexidine gluconate oral rinse, but a much stronger chlorhexidine gluconate preparation, which is, in this particular case, we tested it against ChloraPrep. ChloraPrep is a pre-surgical skin prep, a strong one, which is used in hospitals. It has 17 times as much chlorhexidine gluconate as the oral rinse, because the oral rinse is only 0.12% chlorhexidine gluconate, whereas the ChloraPrep is 2%, 17 times as much chlorhexidine gluconate, and it also has 70% isopropyl alcohol. So it has two key actives, 2% chlorhexidine gluconate and 70% isopropyl alcohol. Our product was able to inactivate Aspergillus brasiliensis in 1/4 the time than that ChloraPrep was able to do. So if there was such a differential between ChloraPrep and our product, then you can understand that there's even a much greater differential between chlorhexidine oral rinse and our product. And in fact, for those of you who do have a screen available, you'll be able to see on this slide that the vial on the right, which contains povidone iodine, is a dark brownish color, a yellowish brown color, and at the same concentration of molecular iodine, that our formulation is actually clear. So there's a big difference between that product and also the ioTech product. Now here, there's in fact, when we also actually tested it. I think I had mentioned to you before about my interest in rhinovirus or in viral pharyngitis from the conversation I had with the ZNT surgeon. And so since a rhinovirus, which is also a non-envelope virus, extremely difficult to kill, rhinovirus is the principal causative organism for viral pharyngitis or sore throats. And we tested Listerine, we tested Scope, we tested Colgate Total. All of them were ineffective against rhinovirus. This is in a 30-second exposure. Whereas the ioTech product completely inactivated or completely destroyed rhinovirus in that same time frame. When you compare the ioTech rinses to other oral rinses, and we looked at chlorhexidine gluconate, we looked at povidone iodine, we looked at chlorine dioxide, 2-part activated, we looked at other chlorine compounds, and in fact, we looked at sodium chlorite. And when we look at the duration of use, chlorhexidine gluconate and also povidone iodine are indicated for short-term use only. The iorinse product is used for either short or long-term. It could be used for chronic use. In fact, we advocate its use on a daily at-home regimen, and for as long as someone has their teeth, or for as long as they have implants. The chlorhexidine gluconate and the povidone iodine stain the teeth and tongue. Our products are non-staining. As far as the effectiveness, for the other products that we compared it to, that both for bacterial efficacy and also for virucidal efficacy, that the molecular iodine products were more effective than these others. It's the lowest toxicity because it's an essential nutrient. We have governments around the world mandate the use of iodized salt as an inexpensive public health measure to help to keep us from developing iodine deficiency diseases. With regard to taste, well, the chlorhexidine gluconate and povidone iodine have an objectionable taste compared to our product, which has a pleasant flavor, either as a mint or as a cinnamon flavor. As far as cost is concerned, the costing of these other products run anywhere from more expensive to 4 times as much. So our product is much more cost effective. In the case of chlorine dioxide products, they were 3 times as much. As sodium chloride products, twice as much, and chlorhexidine products for the same equivalent volume of product, they were 4 times as much. Chlorhexidine gluconate is irritating. It's also a potential carcinogen. If you look at the material safety data sheet on your chlorhexidine gluconate oral rinse, you see it's a potential carcinogen. It has 11.6% alcohol, so it's irritating. And it's also been implicated in rare but fatal anaphylactic shock. And as an additional side effect, it also shows the development of dental calculus. The iodine preparations are ready to go. The molecular iodine preparations, yet the chlorine dioxide, the 2-part activated, requires mixing it for each use and then waiting for 30 to 60 seconds. As far as the duration of effect, nothing lasts as long as the molecular iodine. There's been studies which have shown that the molecular iodine has lasted 4 times as long as chlorhexidine gluconate on microflora in the periodontal pocket. And so when we're talking about efficacy, you're talking about safety, you're talking about cost comparisons. This molecular iodine is fabulous. And in addition, it's important really to note, not just with our iodine, but with any iodine, that iodine does not allow the development of bacterial resistance. I mean, they name bugs due to their bacterial resistance. VRE, vancomycin-resistant enterococcus, MRSA, methicillin-resistant Staph aureus. It just, the list goes on and on. So most antimicrobials and antibiotics allow microbial resistance to develop. That does not happen with iodine. In fact, not only is there no evidence in the literature that microbial resistance develops to iodine, but iodine, molecular iodine, is so potent that our formulations have been used to completely inactivate Bacillus subtilis spores and also Clostridium sporigine spores, which are the spores which are designated by the FDA for spore test strips to validate the efficiency and efficacy of autoclaves, to show that they actually are sterilizing. Because if you can kill Bacillus subtilis and if you can kill Clostridium sporigines, then as far as FDA is concerned, then you can kill microorganisms and can be classified as a sterilant. So across the board, it's extremely effective and extremely safe. So it's a boon for many different applications in dentistry, and those we expect to bring on board in the future, including anti-caries rinses, creams for viral ulcers, antifungal rinses for the mitigation of caries, plaque, and decalcification for ortho patients, for pre- and post-surgical rinses for dry sockets and incision site infections, as an intraoperative irrigant for ultrasonic scaling and root planing, for endodontic irrigants and sealers, as a antibacterial dental scrub prior to bonding of restorations and cementations of prepared teeth, and for pre-surgical scrub, for surface disinfection of impressions, instrument disinfection. I mean, the list goes on and on and on. It's a wonderful breath freshener, and also as a solid surface disinfectant. So there's a lot of opportunity within the professional dental arena, and I'm sure that's going to keep us busy for a very long time.
So are dentists buying this product on your website, iotechinternational.com? Are they buying it right now?
Yes, the ioTech product available on we have two portals on the website. One is for dental patients, and the other one is for a dentist, which is password protected, and it's for dental offices only, and where dentists can get wholesale prices. And the products available are the ready-to-use mouth rinse, and that's 100 parts per million of molecular iodine, which is about 30 to 50 times more concentrated than that which is in povidone iodine. And we have a concentrated irrigant to be used with an oral irrigating device. We've had it tested on with a water pick with their pickpocket tip, and that's to be used as a medicated rinse at home as an irrigant for subgingival irrigation. And we also have a gel, which is 250 parts per million IO-gel, which is to be used in full arch trays as a periodontal tray, much like a whitening tray. And that's for more severe cases where patients would wear it 15 minutes a day. So that's another product that's available on our website. And so there's basically three different products that we make available. We've had these products initially that we had a pilot study done 3 years ago for these products, and it was done by a board-certified periodontist, and with 53 patients. It was over a 6-month period of time, and the results were dramatic. They were compared to chlorhexidine gluconate. They were superior. And in fact, it gave us the impetus to develop our first products, professional dental products, to be used in this area. So after that, and by the way, the patients who were in that 6-month study, to this day, most of them are still using this product on a daily basis because the periodontist doesn't want to take them off of it, and the patients don't want to go off of it. We recently had a study done, or an evaluation done by Gordon Christensen's clinician's report, and that was there were two evaluations done. One was for the ready-to-use rinse with 125 patients in 25 dental offices. The other one was for the concentrated irrigant, and that also was a separate 25 dental offices and a separate 125 patients. And the results were so good in that clinicians' report voted both of those products as best products of the year for 2019 in their annual buying guide. So, the ingredients in these products, not only are they essential nutrients, but they're also on the FDA's GRAS list of generally recognized as safe substances. So they're natural, organic, and so yes, that's what's on our website.
So Gordon Christian, his wife Rella has a PhD in microbiology. What does Rella think of this?
You know, I haven't spoken directly with her about it, so I can't answer that. You'll have to talk to Rella about that.
Are any of the periodontal schools using this right now? I mean, how many, what is there, 56 dental schools in the United States? How many of them have a perio program, and how many of them are going to start trying this?
Well, actually, we were actually waiting for that clinician's report evaluation to be completed, which was just recently evaluated, and so now we're first like introducing this product, and in fact we just now started our marketing campaign, so it's first now being introduced. We've sent product to several different dental schools. And there are a number of periodontists that are using it in the United States. Ziv Mazur, who's the president of the Israel Society of Oral Implantologists. He was a previous president of the Israel Society of Periodontists. He's been using it in his practice, and he lectures in the States a lot to periodontists around the country. He was a keynote speaker at the American Academy of Periodontology. There are a number of opinion leaders who are beginning to use the product and in their offices, and also starting to talk about it on a lecture circuit. So it's just now being introduced.
And what about again, I just love that column 17. I mean, fluorine, chlorine. I mean, dentistry use chlorine dioxide for bad breath. Have you tested this against aromatic sulfur compounds to see if it also treats malodor or bad breath?
Yes. In fact, yes, we've used it with the halimeter, and yes, it's very effective. In fact, our formulation, we've tested it, and it provides long-term benefit for these volatile sulfur compounds. Long-term benefit because it's the most effective in killing the bacteria that produce them.
I think it's interesting out here in Arizona how people don't understand the periodic table because if they have a swimming pool and for some reason they decide they don't like chlorine, they pay to have their whole system in place, which is just bromine, which is just basically the same family right underneath chlorine, fluorine,
right.
Chlorine, bromine, and it's amazing how iodine was been treated to treat goiter, iodized salt. I mean, it's just an amazing classification of elements. What about people who would think, well, I'm allergic to shellfish, I can't have iodine? What would you say to that person?
Yeah. So there's probably 2 or 3 main concerns about iodine. One would be a shellfish allergy, and that's pretty prevalent. But the shellfish allergy turns out to be a sensitivity to a protein in the muscle of the shellfish. So in the literature, in the medical literature, it's replete with references now to that this is not an iodine allergy; it's a protein sensitization. So that's not an issue. There's also there's a concern with iodine as to thyrotoxicity, and we see this from time to time where some of the research has already shown that the thyroid gland selectively uptakes iodide. It does not selectively uptake molecular iodine. So thyrotoxicity is really not a concern because there's no greater, when even if it was ingested, there's no greater concentration of molecular iodine in the thyroid gland as there would be in any other tissue of the body. But our products are not intended for ingestion, so that's really of limited concern. The other concern that people have would be with this iodine contrast media for imaging, and the distinction here is that yes, it's true that these are iodine-related contrast media, but in the first instance, it's being injected directly into the vein. So you're getting a shock to the system. There's a large bolus of fluid being injected into the vein. So the quantity of iodide which is being delivered is far greater, and it's also iodide. The second part is that it's also been found that there are excipients in the radio contrast media, and those excipients which people are developing a reaction to, and these excipients, particularly those that have high osmolarity, have engendered the greatest reaction. So when they've using the same amount of iodide, substituting the excipients, the low osmolarity excipients, they found that the reactions fell off dramatically. So, this is having a reaction to some iodine contrast media is not the same as having a reaction to iodine, particularly molecular iodine. And again, our products are not intended for ingestion, so this just topically applied. There is some absorption, but nowhere near what you would get with ingestion. Also, there had been a study which had been done on over 2,000 women in the United States. This was for the treatment of fibrocystic breast disease, which is a precursor to breast cancer. And molecular iodine tablets were used daily by these women for over 2 years. And there were no reportable adverse events in over 2 years, and the amount of iodine that they were ingesting, I'm talking about molecular iodine now, the amount of iodine that they were ingesting is probably 10 to 50 times as much as what would be available in a bottle of our product, if our product was to be drinking, which it's not.
So you think in the developing world, I don't like to call it the third world, I think that's kind of rude, but there's 238 countries. Do you think where they have a problem with goiter, that this absorbable form of iodine, molecular iodine, would be a treatment for goiter?
It could be a treatment for goiter. Actually, it has a much more profound, because there are other things that could be used for goiter. This actually has more profound use for third world countries, and we're straying outside of dentistry, but we're also again working with large corporate partners. We've looked at this in agriculture, and the statisticians are indicating that in 50 years from now, there's not going to be enough food in the world to feed the world's population. There's just not going to be enough food. So right now, the produce, the vegetables, the fruits, the beings produced, at least 40%, maybe 45% of all that produce spoils before it's ever eaten. Spoils before it's ever eaten. So that if there was a way of reducing the amount of spoilage or increasing crop yields, then that would go a long, long way in order to help to feed the world's populations and helping people in these third world countries. And so to that end, on a pre-harvest basis, our products, I think I had mentioned to you that iodine does not develop or allow microbial resistance to develop. Well, the fungicides that are being used now in plants are, many of them are like neurotoxins. And the way they even know that they're in use, sometimes the birds are falling out of the sky. So to have something which is so safe as an iodine, which can kill the fungi and bacteria that are responsible for, and viruses, which are responsible for crop destruction, and do it so safely, is something which is very, very important to large agrochemical companies, as well as the idea that since no resistance develops, and it does develop to other fungicides and pesticides, within 2 years of a new pesticide being introduced, those pesticides, these bacteria and fungi begin to show resistance, and they start to lose their effectiveness. these agrochemical companies are spending tens of millions and maybe even hundreds of millions of dollars to bring new products to market, and then finding within the first 2 years that after they've got it registered in all the countries that they want to use them in, that they're no longer being effective. So to have something where resistance is not a problem, where it's safer to use, where it's more effective, is a boon. And in fact, to that end, there's a ringworm which is called a nematode, and these nematodes are actually cause more destruction to crops than all the insects in the world combined. There's $157 billion in crop loss each year due to nematodes, and this runs the gamut, all kinds of crops, whether they're grown in cold weather climates, tropical climates, doesn't make any difference. So they found that, in fact, the Mexican Agricultural Institute, which is funded by the Mexican government to help to keep Mexican farmers technologically competitive with other producers in the world, because most of their production comes to the United States. Then their laboratories tested our products, and they did it for nematodes, and they found that ours was the only. Not only was it superior to the other nematicides on the market, but actually it's superior in the sense that it killed nematode eggs. It was the only one that they've tested in their laboratories in 20 years that was able to destroy nematode eggs. It's really an enigma how, on the one hand, you have something which is so effective, and on the other hand, is so safe for human consumption. So safe. So that's for crop yields, and also for freshness extension of crops. They also found that because our products, this is post-harvest now, because these our product also helps to destroy the spoilage bacteria and spoilage fungi on fruits and vegetables, that these crops last longer. And in fact, we've gotten a 2 and 3 week extension of shelf life on different berries and fruits compared to not having been treated with our product. So it has huge applications within agriculture, and also, in fact, for all perishable foods, fish, meat. It sounds like a miracle, but every time I look at it, it's amazing what this stuff does. And that's why, compared to all the technologies that I've been associated with, this thing is so ubiquitous that it has so many different applications, and some very, very important ones are in dentistry, but there are other ones which are going to help mankind outside of dentistry as well.
Well, Herb, you know what I wish you would really do? You're talking about, you're on this podcast talking to dentists as they commute to work. If I go to Dental Town, there's 250,000 dentists. They posted, oh my gosh, I don't even know how many times have they posted. But anyway, when I do a search for the word iodine, there are, how many threads come up? There are 90 threads talking about iodine. I wish you would go to Dental Town, and that's why I love the message board over like Facebook or Twitter. I mean, Twitter, let's say that you were talking about something a year ago. Well, I don't really have a way to search that to find out what, to find that conversation. But on a message board format, you can just do a search for iodine. And I wish you would pull up all those threads and find a good thread for you to enter at and start sharing these slides that you put up on the today. You can post those images in there. You can even post a YouTube video or any video format. But let's continue this discussion on the message boards of dentaltown.com.
Absolutely. I look forward to that, and also I want to work with you folks to help to develop some continuing education seminars as well. Yes,
we have over 450 online CE courses that have been viewed nearly 1 million times. I think the millennials like taking CE at home an hour at a time instead of flying across the country for these weekend extravaganzas. But Dr. Herb Moskowitz. Go to his website, check it out. It's ioTechInternational.com with Herb Moskowitz. Herb, thank you so much for coming on the show today for an hour and educating my homies on all things iodine.
Howard, thank you and your audience for your time.